on temperature or UV today, so you don't need to worry about your notebook quite now on that, but turn to where your unknown project notes are. Unknown project. Everybody open that up. Open that up, guys. I just want to make sure we're all where we should be on that. Okay, so hopefully you didn't like just write it on some like cocktail napkin and then leave it in your coat pocket or something. You actually wrote in your notebook. So look in your notebook, guys. Don't look at me. Look in your notebooks. You should have a mixed gram stain. So look in your notebook. Don't look at me. Look in your notebook. Look in your notebook. Under the lab that says bacterial unknown. Do you have 2 bacteria in there? Who has 2 bacteria in their gram stain? Mixed gram stain should have had 2 bacteria. And I think I told you the next day that hint, hint, wink, wink, you should have had a gram positive and a gram negative. Now, you might not have seen that the first day, and that's okay. But you should have something drawn in there, okay? So when I was grading notebooks, I didn't really grade that, but I did notice, I don't know, maybe half of you hadn't even written that in there. So you want to do it as we go along. That's the whole point. When you work in a research lab or a medical lab, you got to, in real time, keep your data. That means you got to record it as you go along, okay? Because every day you're going to forget more and more. So hopefully you have something in there. So how many of you have something written here? A gram stain, a mixture, okay? Then you should also have 3 plates that are nutrient agar plates in which you streaked out the mixture of those 2 bacteria on the very first day. And remember, you're supposed to look at all 3 plates and try to find 2 colony types. Okay, do you remember that? So you should have drawn what you saw on here, and then you should have tried to say, okay, colony type A is, for example, opaque and big and white and furry and dry. I'm just making up words here, okay? Colony type B is transparent, wet, small, colorless. I'm making up terms. I don't know what you got, but you should have recorded somewhere in your notebook what you thought the 2 bacteria looked like. How many of you actually did that? Okay, not all of you did. But you were supposed to write down what they looked like. Okay, that's called the cultural characteristics. One of the cultural characteristics you should have written down was what you thought was the optimal temperature for colony type A and colony type B. Okay, you might be wrong, but at least you should have tried that. So hopefully you did that, made some attempt. And then, remember, what you did was, hopefully, and I see it for most of you, you should have tried to do a gram stain of those 2 species, okay? Species type A, a gram stain, species type B, a gram stain. So you could start to get an idea that, oh, that big white furry one is a gram positive rod, for example. Or that little transparent one is a gram negative rod. Again, I'm making this up. I don't know what your particular mixture is. So most of you have at least 1 drawing here. How many of you have at least 1 drawing in there? Good. Okay, so if you don't have any of this stuff, today's the day to get it. Okay, you should have saved your gram stains. Okay, so you have at least 1 of these. Okay, and then we did one other thing. So again, I'm just talking while our temperatures are getting up there. We also took from the McConkey and the salts. Guys, listen up. So how many of you had growth on a McConkey? Okay, so I don't know if you remember this. If you had growth on the McConkey, I told you to take a small bit of that and get it growing on a slant. Do you guys remember that? Okay. So if it's growing on the slants and it came from the McConkey, what gram reaction is it?
Negative.
Negative. Okay? And a few of you had your gram positive. positive that grew on a salt. You remember that? There was a few of you. How many of you had that? Okay, some of you did. Okay, I know I gave a few. So if you had it growing on salt, you were supposed to do the same thing. Because guess what? If it grew on salt, what was it? Gram positive. So all of you should have one slant that says your unknown number and that it came from the McConkey. You know now that that is whichever of these two species was gram negative. Because if it grew on McConkey, it has to be gram negative. Okay? So I believe all groups had growth on McConkey, and you put it onto a slant, that is your gram negative. You should have that slant. A few of you also had some growth on a salt plate. You should have labeled that slant, your unknown number, and that it came from a salt plate. Now you know that's your species that was the gram positive. So what we're trying to do here in all this is we're trying to separate your gram positive and your gram negative, get them growing on slants. So what we're going to do while we're kind of waiting for things to dry here is we're going to make, yet again, 2 more gram stain smears. Only now you should know a little more, okay? So all of you need to gram stain your slant. That means take a small pinpoint or invisible amount and make sure what you got on that slant is what you think it is. Okay, so what do I mean what you think it is? Well, if the slant came from McConkey, what should the gram reaction be? Gram negative. If it came from a salt, what should its gram reaction be? Positive. Positive. So you're going to confirm that from your slants. Okay? So listen up, guys. If you put it on a slant, you're going to gram stain it. You're also, guys, going to try to find the one you're missing. Most of you are missing your gram positive, correct? That means your gram negative is already growing on a slant. You're just going to confirm that today. Most of you should have a gram positive somewhere out there on your plates. You may have gram stained it last time, but we're going to try to get it growing on a plate all by itself. So guys, look up here, guys. If you have, for example, species A is a gram negative, that's the one that you should have on your McConkey. And you're just going to confirm it by gram staining the slant, okay? But if species B is a gram positive, today you're going to take that species B colony type and streak it for isolation on a brand new plate. So it's growing on the plate all by itself. Because what we're going to try to do is get each of the bacteria growing all by themselves on a plate and then transfer them to a slant. So today you want to confirm your gram stain of whatever you got on the slant. So you're going to get your slant. The slants are all at 30 degrees. You're going to gram stain that and try to do a good one and make sure it looks like you think it should. How many of you actually saw your gram positive on the plate, but it didn't grow on McConkey, okay? So, and you already have it, you already know which colony type is your gram positive. You're going to take a teeny bit of that colony and you're going to streak it for isolation on a new plate. So I'm going to come around and eyeball people's plates and slants and make sure you understand. So what are you all going to do with your slant? Gram stain it, just to make sure it is what you think it is. From your plates, however, you're going to take a small amount of your missing one, which is probably your gram positive, and you're going to streak it for isolation on a new plate. I'm going to come around and look at your plate. So you got to go get your stuff. While, again, we're waiting for our water to get stable at that temperature. While we're waiting, we're going to get some smears drying. We're going to look at our plates, okay?